So this is the process of replication. Here, the entire DNA molecule will be duplicated and replicated. That means here, so two DNA strands will be separated, and these two DNA strands will be used as template for the formation of daughter DNA strands. But here, in this process, this is a transcription process. In this process, the complete DNA will not be completely rewind. The DNA strand will be released, and here it is forming mRNA. And these are the differences we have to identify. We have to know that the first one is a definition part. So definition of DNA replication. Here, this DNA replication will be useful for formation of new copies of DNA. That means a daughter DNA will be produced by the process of replication. The transcription process here. So here the DNA information, the hereditary information will be copied, and here mRNA will be formed, and this DNA copy so will be reaching to the cytoplasm area. So here there is no formation of new DNA copies. Here RNA copies will be produced and it will be reaching out. And significance. So here DNA replication will be useful for division of cells, the process of mutation here. So this replication process will be useful for formation of daughter cells and for preserving the DNA. So why? Because DNA will be must be preserved for the survival of the species. So if DNA replication is not happen, so the survival is very difficult. And the transcription, the significance of transcription is useful for the gene expressions. So that means genes will be present in a nucleus, and which will be released out in the form of mRNA, and that genes will be useful for the formation of proteins and enzymes, etc. So that's why this is useful for the gene expressions. And here genetic information will be transferred from DNA to DNA, the parental DNA to daughter DNA. But here genetic information will be transferred from DNA, that is parental DNA, to mRNA, so messenger RNA. And DNA replication will be occurred in S phase of cell cycle. So during the time of mutation, during the time of separation cell, and transcription will be occurred in the phase of G1 and G2 phase of cell cycle. And here the main motive of DNA replication will be a cell division. So during the time of cell division only DNA replication will be occurred. And here the main motive is for the synthesis of protein. So for synthesizing proteins, so we need transcription process. Here DNA replication will be used in cell division, and here it is used, transcription will be used in gene expressions, that is protein synthesis. In DNA replication, so we are using DNA polymerase enzyme, so which is useful for the DNA replication process. So that DNA polymerase will be useful for formation of a polymeric chain, which is for the elongation of replication. And for elongation process, it needs some nucleotides. DNA polymerase 3 enzyme, so which is having these nucleotides, that DATP, that is deoxy ATP, deoxy GTP, deoxy TTP, and deoxy CTP. These are the raw materials for the elongation process of the DNA replication process. But whereas in case of here transcription process, so ATPs, we are not using DATP. So why? Because here we are forming RNAs, we are not forming DNAs. So that's why we are not using DATPs. Here we are using ATP, UTP, CTP, and GTP and CTP. And here one more important point is, here we don't have TTP. Why? Because this is RNA. So in RNA, so UTP will be present. That is uracil triphosphate will be present as which are used as raw materials. And here in the DNA replication process, both DNA strands will be templates. Both parental strands will be templates. But here, so only one strand will be template. Another strand is not involved. So one strand only involved for the process of formation of mRNA. Only single strand will be used. And here in DNA replication process, we required primer to initiate the process, to initiate the replication process. But here in transcription process, we doesn't require any primers. So we need factors and RNA polymerase enzyme at the promoter area. we need a Tata Boss sequence, we need a -35 sequence. And here enzymes used in DNA replication is DNA helicase. So DNA helicase will be useful for separation of parental strands, and DNA polymerase, so which is useful for the formation of daughter strands. And here in transcription process, we are mainly using RNA polymerase. So RNA polymerase will be useful for the formation of mRNA. And here the unwinding. So unwinding of the helix will be occurred entire DNA molecule. That means entire DNA molecule will be unwinded, and we are forming two parental strands here in DNA replication process. But here transcription process, the unwinding of genes will be temporary. That means a small sequence, a functional gene unit will be unwinded, and there at that position, RNA polymerase will be added at promoter region, and it is useful for the formation of mRNA, and finally it will be detached. That means the entire DNA will not be involved in this process. Only a functional area, a functional sequence will be used, will be involved in this process. And here in the DNA replication process, adenine pairs which are attached with thymine by the hydrogen bonds. Adenine is attached with the thymine by two hydrogen bonds, and guanine is attached with cytosine with three hydrogen bonds. But here, so adenine is attaching with uracil. So why? Because here thymine is absent. So why? Because we are preparing mRNA. So in RNA, so thymine molecules will not be present. So adenine is attaching with uracil. So here in the DNA replication process, the entire template, entire parental strand will be copied. But in transcription process, only genes, a functional gene sequence will be copied, transcribed. So not entirely DNA will not be transcribed. Only a gene sequence will be transcribed or copied. Here the final products will be two daughter DNAs in DNA replication process. But whereas in case of transcription process, here the final products will be mRNA, sometimes mRNA, tRNA, rRNA. mRNA will be formed by the enzyme of RNA polymerase 2, and tRNA will be formed by the enzyme of RNA polymerase 3, and rRNA will be formed by the enzyme of RNA polymerase 1. So these are the final products, and non-coding RNAs like microRNA also will be the final products of transcription process. And here in DNA replication, so we are forming a double standard DNA. So double standard DNA, that means we are producing two daughter DNAs which are having a double standard. So one strand from a parental strand and one is daughter strand. That is a semi-conservative model we have already discussed. And here we are getting only single stranded RNA. So RNA will be always single stranded. So here one single stranded RNA will be produced. And here in DNA replication process, so we are forming Okazaki fragments. So yesterday we told about these Okazaki fragments. So these fragments will be forming at lagging strand. So after that, at the end of the process, replication process, these Okazaki fragments will be attached with the by the enzyme of DNA ligase, and finally forming a daughter strand in the replication process. But here, so after formation of mRNA, here RNA will be edited. So why? Because so mRNA is initially inactive in nature. So why? Because it is a premature mRNA. So now it has to be matured, it has to be activated, and it is going under certain alterations, certain modifications. So then only it is converting into mRNA, a matured mRNA. Then only it will be useful for the formation of proteins in the translation process. And here the process in DNA replication process, we doesn't require any further processes after formation of daughter DNA. So we doesn't require any another further processes. But here we required some further processes for making a matured mRNA. and those processes will be splicing, terminal addition, and base modifications, etc. And here the bond, replicated DNA strand is always having hydrogen bonds here, and which is having two strands. And here, so we doesn't have hydrogen bonds. Why? Because here after formation of mRNA, so which will be separated from the main strand. So here RNA is having only single strand. So that's why here there is no double bonds which are present in that. And after DNA replication process, the products, so what we are getting daughter nucleus, daughter DNA molecules, so will be remained in the nucleus only. It is not going into the cytoplasm. It is remained in the nucleus only. But whereas in the case of transcription process, here after formation of mRNA and a matured mRNA, then it is going to the cytoplasm and it is useful for the further processes like translation. And here in the DNA replication process, the products are not degraded. That here the products are daughter DNAs and these will not be degraded. But whereas in case of transcription process, the products are degraded after its function is over. After its formation of a protein, the mRNA will not be useful and it will be degraded. Rate of production. Here, DNA replication process will be very faster than it is 20 times faster than a transcription process, very much faster. And here it is forming 6 or more replication forks. So we have already told about this for replication forks. So here we are getting more than 6 replication forks for the eukaryotes. So what about in normal cells? Here it is forming only one replication fork. So what about in rate of production of here? It is slower. Transcription process is slower comparing with the DNA replication process. And process followed by here after finishing this DNA replication process, then transcription process will be started. And here after finishing the RNA transcription, then translation process will be started. The final significance of DNA replication is entire genome is conserved, saved for the next generation. Here genome will not be divided. The genome which is present in the parental cell, which is same equal to the DNA genome which is present in the daughter cells. Here genome will be preserved. So that's why survival of species is occurred is mainly due to DNA replication process. And here in transcription process, the main significance is for the protein synthesis. For for doing for many metabolic processes, we require proteins and enzymes. Proteins are enzymes. So these enzymes or proteins will be forming in the cytoplasm. So by the taking the information from the nucleus. So this is about the difference between replication and transcription process. Okay, ma. Stop chain the recording.